then if you take the patients that are refractory to treatment, they don't respond to the therapy, and you take the microbiota from patients that respond to therapy and you give to the patients that did not respond, some of them start to respond to the treatment.
Hello.
Hey team, what you got?
So today I have another friend with me. So we had two of my friends. Maybe we should invite some people at some point. I have a friend with me, Antonio. Say hi, Antonio.
Hey, hi guys.
So Antonio is a good friend of mine, back from New York also. Although we just figured out that you guys never met. Antonio, just like me, is a scientist, or I guess he still is a scientist. I'm not anymore. We met when I was doing my PhD, and he did his postdoc. If I'm not incorrect, is that correct, Antonio? You did your
That's
postdoc
correct.
back then, right? Yeah.
Yeah, as an important.
So, and we both, we're in the field of biology in the broader sense, but he did something fairly different from me. So maybe, Antonio, you can say a few sentences about what you did back then and what you do now.
One intermission here.
Yeah.
Please explain it like I'm five because I'm not from the industry. So anything that you say, if you explain it like simply, I would really appreciate.
Sure. Yeah, I'll try. So um, I'm a scientist. I would say I can extend or make it shorter. So I'll try to make something in between. But if you guys want to extend and ask questions specifically, I can say. So I'm from Brazil originally, and I did biology and mathematics. And my intuition for doing that one is because I thought mathematics was fun up to a certain point until it gets too hard that you have to study to solve the problems instead of solving the problems, and it gets complicated. And biology, I just think it's fascinating. The diversity of biology that we see in the world, the evolution. It was a topic that I was very, very excited from an early stage, and I want to combine both. And my intuition is that with quantitative approach, with the mathematical modeling, with the mathematical thinking, you could frame biology in a structured way that would help to understand better the evolutionary process. That was my intuition back in the days. And then I did a PhD in bioinformatics. Bioinformatics is the area of that integrates biology, computer science, statistics, mathematics together. The short summary is that you're studying the genome of the organisms. I don't know the complexity to explain genome for a 5 years old kid, but genome is uh, is the source of information inside the cell that 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 tells how who you are. It codes the information of every cell in your body, specific features from you. There is influence from the environment, of course. So the genome is not deterministic, but we can we can see one thing, since I mentioned evolution, one thing that is interesting on the genome is that we can track the path from the genome based on it's a sequence of DNA, which is a sequence of letters, and based on what's conserved in these letters, you can see the past, and you can see the past from billions of years ago.
What is that called? Like cryptogenomics or something? Is
I
that
don't
the
know.
name?
I don't know if it's called. Maybe it is. Yeah. Maybe it is.
So
Uh,
basically, like, correct me if I'm wrong, but I think from what I remember when I, when I used to still do science, you basically compare sequences for different genes across different species and try to, by comparing them to each other, infer what the origin gene looks like, and that, thereby reconstructing history. Is that correct?
yeah. Exactly. Yeah, you can do that.
Yeah.
Uh-huh. And using uh, the uh, you can see the geological time of samples and collection to have some idea of to calibrate the clock as well.
Yeah, yeah. Because you
So
know, like the rate of mutations and stuff, right?
yeah, you can
Yeah.
estimate them. Yeah,
Yeah, I remember I watched a talk by one of my other friends. I think it was his PhD thesis, maybe, or something. During his PhD, and I think what he did was he did what you just said. Like they calculated some ancestor gene of some enzyme, and they dated it to, I'm gonna say some wrong numbers, but like I don't know,
yeah.
a billion years. I'm just gonna say a billion years. Who knows? But but then they like estimated the I think they estimated the perfect temperature for it to work at, to do its job, or maybe it was like the melting temperature of the protein or something, that the enzyme.
Yeah.
And they figured out it was like way hotter at, operating at way hotter temperatures than ours. And then they like inferred that probably it's because the ocean temperatures back then were like 90 degrees Celsius and not 20 whatever it is now,
Oh
because it was
wow,
so hot back then. Yeah,
interesting.
yeah.
Interesting.
Yeah,
Okay, so basically, you are you're focusing on. So right now you're studying. What are you
yeah.
researching? Okay.
Yeah, so then this was the past. So I'll make a fast tape. I'll try to make in less than 30 seconds. So then, because I really like evolution, then I start to be interested in bacteria and microbiota. And it's interesting because they are tiny organisms. The generation time from them can be 20, 30 minutes. They can double in a laboratory condition, and you can see the change in the genome and the change in how they adapt in the environment, and also in the change how they interact with each other very quickly. And you can make hypotheses, test them, and experimentally validate. And I was very interested in evolution of cooperation and other ecological roles that happen in bacteria. Even though I never worked with it, this was a topic that I enjoyed reading. I thought it was fascinating. And then because I started working with bacteria, when I went to Columbia University, when I met Tim, I was working with evolution of horizontal gene transfer regulatory regions, which are regions in the DNA, in the genome, that tells the organism when they should express a gene or not. And the way I say that in a more broad sense, it's not only about having the information, but using the information at the right time, at the right context, in the right amount. That's what makes an organism adaptive. And you can
Can you
expand
also
this.
say
Yes.
Can you also say one sentence about horizontal gene transfer? Because I don't think
Oh,
everybody
yes.
understands
Yes,
this.
of course. Yeah. Horizontal gene transfer is because bacteria, they are unicellular organisms, so it's only a single cell. And the vertical transmission of information is when they duplicate the DNA and they become two cells. They divide and they become two independent cells. But they can also exchange pieces of DNA with each other, even if they are very far from each other in the evolutionary path. And this piece of information can help them to adapt to an environment. A common way that you can think about it is drug resistance genes. So a reason that drug resistance genes spread so much in the environment is because a bacteria that carries that gene could transfer a plasmid, which is a small piece of DNA, to another bacteria horizontally, and they the bacteria that receives that piece of DNA becomes resistant to antibiotics as well, which has a medical concern, because if it's a drug resistant bacteria, when you're going to give an antibiotic to treat an infection, they may not respond to the antibiotic, and then you have to give a stronger antibiotics, and there are extreme cases that we don't even have antibiotics arsenal to deal with the infection.
And horizontal gene transfer only happens in bacteria?
Um, it could happen in non-bacterial organisms, and it can happen between bacteria and eukaryotes. For example, the eukaryotes, it's more common to see a piece of DNA in the bacteria, but mostly is in bacteria.
Okay.
Okay, let me pause here and ask you. I have a few curiosities about antibiotics, which I think, am I correct? by thinking that you might have some good expertise on that front.
Yes, yes, yes.
So I have a few. First of all, just an anecdote. Somebody, I don't remember who, few days ago, a few weeks ago, told me that somebody they knew had like a very, a case of like, oh gosh, my memory is at fault here. But anyway, so they had a case of uh, basically they were sick and they needed antibiotics, but the antibiotics they were um, receiving were like classic antibiotics that you buy in pharmacies, and they weren't really working. And so at some point, uh, a member of the like medical staff of the government or something like that came uh, by helicopter with like a black like luggage basically, and like it provided like a class 2 or level 2 antibiotics that are kind of like not available in the market and not even in hospitals, but they're like left as a sort of like last resort in case the antibiotics that we generally use don't work to defeat like the. So I don't know if this was the bacteria. I don't know if this was like a joke or
And they came with a helicopter to your friend's
yeah, kind
house.
of like because it was
Yeah, some people
not
have a lot of
exactly how,
That's
but
resource.
really.
it was yeah, Italy, we kind of like spend money like this. No, I think it was because he was in a hospital, and so the
Okay,
problem
okay.
was that it was very time sensitive,
Yeah,
and so basically they rushed in,
sounds like a movie. Yeah.
yeah. And the thing though, probably it was making fun of me, but it might not be like, is it, do you are you aware of this concept of like class 2, like level 2 antibiotics that like are not available and they're kept not secret but like aside so that like we don't get immune to those as well?
Yeah, I think the helicopter scene was a luxurious that he had. But yeah, hospitals have like it's going to fail the right technical name. I don't know if they call first, second, third, fourth generation of antibiotics, but you do have the ones that are called last resorts, which are they they are usually broad spectrum and stronger antibiotics that you give when the bacteria is resistant to everything else. And that's common, yeah, and that's when make people alarming when you see a highly resistant bacterial type. Then this is what worries most, and it's a race because uh, we develop antibiotics, and then at the beginning it works well, but later the bacteria develops resistance to that antibiotics, and then we realize that it's not going to work. Uh,
And
uh,
these last resort antibiotics, they're kept away from hospitals. Is that correct? Like they're.
no, I'd say the hospitals have a storage of them. It's just they don't use unless it's really really needed to use them.
Okay.
Okay,
They,
so
they
it's really a tier 2 level that like basically is there available for the like special or like super like last resort case. That's very
yeah, yeah,
interesting.
yeah, but
And will
I
they
don't
at
think.
that point? Sorry. Yeah.
Yeah,
Go ahead.
say.
No, no. Will they at that point isolate that patient just to make sure the resistant bacteria doesn't spread to other people, like to like bring him to
Probably,
ICU or
yeah,
something?
probably, yeah, yeah. There's a risk of hospital infection transmission, right? Like a nosocomial infection, which is when
Mm,
they a
wow.
bacteria uh, from one patient or something that is happening in the hospital starts to infect other patients.
Which, by the way,
So
as far as I understood, it's even like makes them even stronger. Like the nosocomial like spread, it actually generally brings like bacteria that is stronger by definition because in the hospital survived. Is that the case or?
yeah, yeah. I not necessarily by definition, but by context, because they are bacteria that are in the hospital and they're infecting people, so they are being treated with strong antibiotics, and then they
Mm,
keep spreading. So the environment that they are exposed, it's the one that has most exposure to antibiotics. So then, if they're surviving that long, it's because they are
wow,
resistant.
that's
Uh,
And
interesting.
also, probably hospitals are like a really fertile ground for bacteria, just because people are probably already weak have a weak immune systems, and they're very crowded, and everyone,
Yeah, but you would argue
Um,
that they were cleaned, cleaner than other environments in a way, right? But yeah, they
Well,
it's a trade off in a way.
I think this is actually a very good point because, and this is part of what I study now, which is the microbiome, the whole ecology and context of the microbiome, and we can get there because you mentioned being cleaner, but being cleaner is not necessarily good for for dealing with a bacterial infection because, and this is where a lot of thoughts have been spent now, which is the best way that you could protect a person from an infection is not giving strong antibiotics necessarily, but having a good microbiota that would um, hinder the pathogen or the opportunistic pathogen to expand. So if you have an environment that is extra clean, let's say extra clean, if you give very strong antibiotics for a patient, you disrupt the microbiota from that patient, and then an opportunistic bacteria has more space to expand and
Mm.
cause an infection
So
than when you have a healthy microbiota.
it's like the example. It's like what we you do with fermented food, right? In a way, like you let some specific bacteria to grow, and they actually hinder the growth of like like bad ones in a way, right? Like, is it the same concept?
The ecology of it, they share similar ecology properties. Uh, so one example, there's one bacteria that is opportunistic and cause infection that is Clostridia difficile, and it was recently renamed to Clostridioides difficile. We call it C. diff,
Hmm.
and it's an opportunistic bacteria that people who have it have a very strong bowel disease and infection, and it kills some people. I don't know by had the mortality rate, and the traditional way that people treat it is by giving strong antibiotics. But then the recurrent rate of this infection is very high. So about 70-80% of the patients is going to have a recurrent infection within a year. The numbers that I'm giving might not be precise,
Recurring
but
infection means that it wasn't really extinguished from the body completely, right? Was it's that the case, or?
yeah, pretty much, yeah. So you treat, you reduce the burden of the infection, so you the symptoms are over, but you still have some spores of the bacteria in the intestine
Hmm,
of that patient, and that spore can regrow and cause an infection again.
interesting.
Let me just say one thing to maybe clarify, and also, maybe I say it wrong, then you can correct me. But the way I imagine is that your intestines, but basically any place that is a place for microorganisms to grow is like, usually, if there's no special treatment with antibiotics, usually it's like full of different types of microorganisms, right? Like they're all competing for the same resources. So like, let's say in your intestine, it's like a jungle full of like micro
Yeah.
organisms, right? And they're all competing for the same resources, and you have like your I mean, maybe it's also something we should define for people who are listening who don't know what it is, but microbiome, can you say just one sentence like what microbiome means?
So in the microbiome is the set of genes, metabolic capabilities of population of microbiota, and then
In your
human
intestines?
microbiome. The
Ah,
human
okay, got it.
microbiome and the gut microbiome would be in the human or in the gut in the intestine.
Okay. Okay. Usually we mean the gut one, I guess, right? When we
Yeah,
say
yeah.
microbiome.
Usually you mean the
Yeah.
gut one.
So like there's like, I think there's a number I heard recently is that like we have the same number of cells in our microbiome that we do like body cells. Is that correct?
Yeah. Depends if
That's,
it's
I mean,
before or
that's
after you
crazy.
go to the bathroom. Yeah, I'm joking.
But like but
Plus
basically
or
you have, so like basically we always say like we as people are like a set of eukaryotic cells, but really like we're a set of eukaryotic cells plus the same number of bacteria that
it's
also
about
belong
that.
to us.
Yeah.
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 And so basically it's like this jungle of bacteria and other microorganisms that always are competing with each other, and if you kill all of them, then there's like, it's like an empty grocery store, and the first person you send in, they will like eat everything and multiply like crazy, right?
That
Yeah.
is why
Yes.
when you take antibiotics, you're kind of like suggested to take lactic ferments as well, like to kind of like uh, help inhabit the right way, because antibiotics would actually kill the most of bacteria in the in the guts as well?
At that about that, you have different type of antibiotics. You have some that are broad spectrum and some that are narrow spectrum. So the broad spectrum, they they are they nuke everything there. And I don't know if that's the proper word to say, but like
We like it. It's
they
a 5 years old word. I like it, so it's good.
yeah, okay, yeah. So they destroy most of the bacteria. And if you use narrow spectrum, it's not as devastating, but it still target the pathogen. And
Okay. Yeah, I'm
Yeah.
fascinated by antibiotics in general and like how they interact with the body, because in a way, we know how they kind of like changed medicine when they were first discovered and introduced.
yeah,
But I was talking the other day with someone who is actually designing clinical trials and making sure that they are, like, done correctly when research happens. And and she was saying that basically nowadays very few institutions are actually researching new antibiotics. And so that's a problem because, as we said before, like, we're getting kind of like resistant to, I mean, at least that's what she was saying, right? We're getting resistant to antibiotics, so we have fewer and fewer available that actually like help in nuking whatever bacteria, negative bacteria we have in our bodies. And by widespread use, what happens is that they get resistant to these antibiotics, and so it narrows down the list. We have a lot of antibiotics available, but it narrows down the list that actually is useful for some of them. And so she was saying that that's actually a concern. And it's strange because, probably because they're cheap, I'm not sure, but like, I don't know why companies don't actually research. Like, this is becoming an issue, right? And I guess it
yeah.
takes time.
Yeah. There are two things to say. One is the traditional antibiotics research, and the other is the novel ways that we can approach to target bacteria. And first, what you're saying is the traditional antibiotics research development. I would say the key reason is the return of investment, because it's very low. It gets more and more expensive to develop something new, but for 90% of infection, you don't need that less class of antibiotics. It works with the arsenal that we have. But then, so you, even if you make something that is going to be new, strong, unique, uh, the costs for development and the return of investment that the farmer field is going to take, it doesn't pay off in general, and that's why the investigation hinders, because you have to pay research, you have to pay clinical trials, you have to pay the drug development, and those things cost a lot of money. It costs billions of dollars, and people make decisions based on what they can be paid off, right? So when like we can do things
Right.
on because we are passionate and you want to cure lives, but at some point, uh, if you cannot make your living out of it properly, and if you don't have the full infrastructure for that, it's hard for you to stick there for a long time.
I'm
Uh,
gonna I'm gonna
yeah,
hypothesize that the big pharma companies, obviously they're profit driven, but if ever the day comes that there's a strain of a bacteria that is basically resistant against everything, that they are aware of the potential profit of being the company that has some new antibiotic that still works, and maybe they keep it hidden. I don't know, but like, because if they are the only one that works, they will become super rich, right?
it could be, but I don't think that's how things are in practice. They develop, and then when they come up with something very new, uh, it often, the return of investment is not that good, but it's possible. But what people have been doing now is looking for alternatives uh, for the traditional antibiotics pipeline. Well, one aspect that you see research, at least on the academic level, is uh, specific antibiotics. So people are taking advantage of machine learning, AI, and also the repertoire of genomes that we have out there. And those genomes help you to predict potential bacteria that produce secondary metabolites that could be used as antibiotics against different bacteria. Because the microorganisms, when they're competing with each other in the environment, they produce small molecules that target another bacteria, and that will give them a competitive advantage. And if you can identify those molecules, that those could be antibiotics, but they function as antibiotics. But the advantage of this is that they could be very specific for a pathogen, for example, and people look for those antibiotics, and it has an advantage. Instead of having a broad spectrum and kills all of the bacteria, if you can have something that kills exactly the pathogen, or just the pathogen and maybe a few similar bacteria, but it's very targeted, you could heal an infection and have a less disruptive impact on the microbiota from the patient. Because this is a topic when you work with human microbiome, is that there's a concept called dysbiosis, which we all have our natural microbiota, a healthy microbiota. It's not a clear defined. There's no like this is the healthy, this is the unhealthy. There's a lot of diversity. That depends on your diet, your geography, um, 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 the region, like your family history or so. Like you could have different impacts that create some variability of your microbiota, but you have a clear distinction between the healthy and the dysbiotic microbiota. So the dysbiotic, the simple characteristic is that the healthy microbiota is diverse, and the dysbiotic is low diverse microbiota.
Can I ask a question?
Yeah. Mm-hmm.
So, how much does an individual's microbiome change based on circumstances? So, for example, something I've been thinking a lot, and that was my assumption, but I don't know if that's true because I've moved countries a lot, that suddenly my environment is very different. Like the water I drink is different, the food I eat is different. Does that affect my microbiome a lot? Because I do feel like my health sometimes change a lot when moving countries, and I don't, could be because of many things, but how much does that actually influence my microbiome? And also things like what I was thinking recently, what if I am single versus I have a partner, or I change partners? Like, does that, just the fact that I'm intimate with a new person, change my microbiome? Things like that.
Yeah, yeah, yeah. So some things I'm not going to be able to be precise and say it does at that level or not, but it does. But I can give an example. There is a population. They are hunter and gatherers. They study their microbiota, and what is interesting studying this type of population is because
Like you're saying, like in the Amazon rainforest or something.
yeah, this was in an Africa
Okay, okay,
population, but they have one
okay.
in Venezuela that they studied as
Yeah.
well.
So
And what
that's kind of like an isolated tribe that has no contact with civilization, or
they probably do some, but their lifestyle are still very traditional. So what's interesting is because we evolved, our whole body co-evolved with the bacteria in our body with that lifestyle. It was only in the last 100, 200 years that we really changed abruptly a lot of how you eat, processed food, antibiotics exposure, and those would have something that is more adapted to our body. And this population specifically, they have their diet changes according to the season. There's one season, the example that they gave, that they eat a lot of honey because there's, let's say, the honey season. It's available for them, and another season they eat more meat, and you can see the microbiota change within the patients when they follow the samples longitudinally. You can see the change that happen when they are in each season, and it kind of oscillates, it returns to a state. Another one, like high fiber diet,
But how
Uh,
much does it I don't know if you can say any numbers, but how much change are we talking? Is it marginal or is it like huge changes?
uh, it's I would say it's huge change. Depends how you define huge change. But because now I'd have to refresh my mind reading the paper again. But it's something.
Like
It,
basically looking at it, I mean, just to give a without a number, but like if I look from one person to the next, presumably there's a big difference between person A and person B. But if within person A the change from season to season is bigger or as big as person
yeah,
A to person
yeah,
B,
I
I
think
would
that's
call
a
it
good
good.
example. Yeah,
And I guess
yeah.
I guess it's also related with. I mean, we know that diet plays a huge role in your microbiome, right? Like what you eat and how frequently you eat it, kind of like changes the microbiome. Like this whole concept of probiotics, which is like getting more and more popular because people are getting like more focused on this stuff. Yeah, I'm always fascinated by it because, in a way, so the theory when you go to the doctor tells you, okay, you're taking antibiotics, take some probiotics alongside it, so that like it kind of like reduces the impact of like this actually not alongside it, but like right in a different time of the day, because if you get them at the same time, it's worthless because the antibiotic kills them all. And then you should continue after you taking the antibiotics for like a month or so. But then I was talking to another doctor, and doctor says, yeah, but most of the probiotics that you buy even in pharmacy, like they're over the counter, they have a very limited amount of the variety is minimal, and they have a very limited amount in terms of quantity. And so there are some like stronger, let's call it medicine, stronger probiotics that you can buy that actually like have a big impact, that like I think we're talking in the billions, right, of different bacteria that you can actually ingest, and that can play a role. But then I was figuring, because I always try to optimize, maximize, etc. Why? What are there like counter, like other negative effect of taking these kind of drugs, which are not dry? Again, they are probiotics, but they're like in huge amounts. If they're generally considered positive, to have like a session like this, like 2 or 3 times a year, to like make sure that to regulate your microbiome.
Yeah, I
Yeah,
always wonder though, because with sorry to interrupt, but like with probiotics, because if everyone has such a different microbiome, how can it make sense to buy over the counter mixes of bacteria if everyone needs very very different bacteria? That's my question. Maybe the answer can be
A lot of
next,
questions,
Antonio.
Antonio. Please bear with us.
yeah, yeah. So the way I'd answer this, first of all, this is a topic of intense research. So a lot of people are debating this. So this is a topic that we're learning more and more over time. But the way I interpret and I think about this, it's about a balance of the microbiota. and and you can think with a medicine, you can say the difference between a medicine and a poison is the dose, right? Which means if you give too much of a drug that would treat a disease, it can be toxic to the patient. If you give too little, it's not going to be sufficient to treat the disease enough, and then you have to find the right dose. And there are plenty of examples to illustrate that this is important. And in microbiota, a similar concept is what I believe that that is important in our body. It's finding a healthy balance of the microbiota. So it's not about and the probiotics, they often focus on a few specific strain species of bacteria that are seen as positive or associated with a positive health uh, outcome, but we don't fully know the impact that they have in the patient. And there are some studies that they are not necessarily going to be better. And a bacteria that is better in one condition may not be better in another condition. And one reason that is a simple explanation, they they the microbiome interacts with the immune system. That's one of the major layer that links the human microbiome with the patient physiology and patient health and disease. And they can make the immune system more aggressive or less aggressive. And if you're in a condition that you need the immune system to be more aggressive, then the type of bacteria that make it more aggressive would be helpful. If you want to make it less aggressive, the type of bacteria that would make them less aggressive would be more healthy, and you have to find a balance. So in cancer, for example, you want your immune system to be more aggressive against the cancer. But in autoimmune disease or other immune disease that is inflammatory, overreactive, overreacting immune system, you want your immune system to be less aggressive. And I think so the point here is that it's not necessarily a good and bad bacteria as people try to define, although there are some bacteria that are highly associated with a health outcome. But and there are some experiments with antibiotics in the immuno-oncology area that if you patients that receive probiotics, they have a worse response than patients that do not receive probiotics when they are under immunotherapy, which shows that uh, the probiotics not necessarily improves the outcome that you expect. It's very complex, and many of it we do by our intuition, but not necessarily with rigor clinical trials.
But
And
it's very
Yeah.
interesting. So you were saying one thing you said um, that struck me was basically you're saying when you have autoimmune diseases, you want your immune system to be kind of like on point, but not too aggressive, too strong, because otherwise that would have an impact on your autoimmune disease. Is that okay? Like, but you don't want it to be too lacking because otherwise you're exposed to external factors, external, right? And so you can actually get diseases more frequently because your immune system is kind of like lower. uh, would that be the case?
yeah, so the immune system has it has both eggs happening together. One that tells this is a signal that you have to be aggressive, and another one that they read and say, no, this is not. We don't have to worry about this. Don't make a big deal. This is just something that is out there that is in the environment, and this is so the way the immune system perceives damage, for example, there are patterns when a cell lies, when the cell breaks, when a cell dies, or even from when bacterial cells die, they release chemicals that could signal the immune system, and that could activate the immune system, or it could or not. And the immune system must be able to understand this is a signal that is harmful for us, and this is a signal that we don't, it's not harmful for us, so we have to tolerate that signal. And the bacteria in the body teach the immune system that, and then it doesn't, it's not that it weakens the immune system, it just teaches the immune system you can tolerate this, don't worry. Or yeah, this is a signal that you have to pay attention because this could be harmful for you. And having this proper balance to distinguish this signal is very important in many diseases.
Can I also ask a question?
Mhm.
I was wondering that before. So let's say I take an antibiotic and I kill all my gut microbiota, and then I, live my life regularly after that. Will I get the same like mix of bacteria afterwards, or will it be completely different just because it's random after that?
Mhm. It's generally similar. So there is a resilience. There's a resistance and resilience. Resilience is the ability to recover to a state that you were before. So to a big extent, the microbiota is resilient. And I was
Like,
working, yeah.
are you saying it's because there's bacteria that survive the treatment, or will they recolonize me from the outside?
They probably the most likely path is that they survive. It doesn't. So think about a forest. if you destroy a forest, if
Yeah.
when people destroy, then after some point they will grow again, right? And then you have the seeds that are there that is going to be able to seed again. So
But then
so
if I
yeah.
take probiotics, then it means it's someone coming in there with like external seeds and like sowing, which it's actually might be counterproductive then, right?
Yes, yes, pretty much. Yeah, I mean, so then you can have that effect. But there is also something that it's a very complex forest, the microbiota. So you have thousands of species or more, and different strains, and they have the ecological niche, how they interact with each other. They are also adapt to the immune system of the patient because there is a education process. The immune system learn about your bacteria, and your bacteria learn about your immune system, and there's a co-evolving process there as well. And this influence uh, the environment and the adaptation. There is one topic when you do bacterial drugs, which you design, and this is not probiotics; it's actually a drug. So it goes through clinical trial, and this is under strong research now. But you want to design a cocktail of bacteria that is going to help a patient outcome in a certain disease. And one thing that is challenging is to see what we call the engraftment of the bacteria. That the bacteria not necessarily stay there. You give the which would be similar to a probiotic, but it's not called probiotic because there is a distinction definition between probiotics and drug. Probiotic is given to a healthy person, and the drug is given to a sick person, and the regulation for that is very different. And uh, and then when you sequence the DNA of the bacteria to try to identify if your probiotic is there, you don't see it engrafting, which means sometimes you see a little bit, but in general, you you don't see the external drug, external bacteria that you give to the patient staying there for a long time.
I see.
It's
Okay,
more
so what?
transient. Yes.
But it helps the recovery of whatever natural bacteria is supposed to be there. Or did I misunderstand?
Yeah, it could help. It helps. It helps to give that new stimulus at that time that it passed through, but it doesn't stay for the long term. So it just passed through, like stays there for a short time, but then it doesn't last long.
Because you
But
don't
it
want it
somehow
to stay,
helps the
right?
recovery of the natural microbiome.
The healthy microbiota recovers constantly. So when you give
Okay.
these drugs, you first give antibiotics so that you open space in the gut for this bacteria to grow,
Mm.
and then you treat the patient. But then later, you when you look at the microbiota, you not necessarily see that the drug that you gave stays in the
Yeah,
patient.
yeah, yeah,
But
yeah.
this vary per treatment and varies per specific disease indication because there's some disease that you actually want the patient to be colonized because it's the colonization that is going to protect from an infection of another
Yeah, yeah.
bacteria, for example.
Makes sense. Yeah.
Interesting. I have two other questions quite pressing on my mind. So one is
Impressive.
if you can um, help me understand what prebiotics are compared to probiotics, because I mean I understood maybe wrongly that it's like basically they are nutrients to the microbiota, like they're actually like they feed bacteria inside our body, and so I wonder if like because that those are we're ingesting with food, right? The prebiotics. So I guess like if there's any like if you don't have the bacteria, although you ingest the right stuff, that's it's kind of like going to waste in a way. That's the first question. If you can shine a light there, and then the other one is about immune. So basically, the relationship with microbiome and oncology, because I think that's fascinating and I know nothing about it, so I'm very curious.
Yes. So the microbiome and immuno-oncology is a part that I have big interest. And the prebiotics versus postbiotics and probiotics. So you have different concepts when you think about bacteria. Prebiotics, it's exactly what you said. They are nutrients that feed specific bacteria. So an example, they are sugars that we cannot digest ourselves, but a specific bacteria can digest, and then it gives an advantage for them to grow. And one thing that happens with us humans and other mammals as well, the milk that we have when you're a baby from our mom, they are full of prebiotics, and this milk it helps to foster the growth of the good bacteria in our body, the common cells that live with us, because they provide some nutrients that they can break down and they can grow really well and help colonizing our gut. And then traditional nutrition thoughts is they think about the macronutrients, the micronutrients, but they often don't think about the microbiota nutrients that you have to feed for a healthy microbiota as well. And now formula for babies, they are starting to consider having these prebiotics as well to match some of the properties of the maternal milk. Probiotics are the bacteria itself. So you give the bacteria for the patient, and postbiotic is you give something that is produced by bacteria. So let's say you grow the bacteria in a lab, and then they produce things. You lyse their cells, and then you take just the extract of the cell, the supernatant that you can get out of it, and then you give the patient. So that would have a complex set of molecules that could have an impact on the patient. Those three are common studied, and of course we mentioned about antibiotics as well. Like this, yes. Yeah.
No, just a question. So you're basically taking the result with postbiotics. You're saying you're taking the benefits of having probiotics in your body to a patient that doesn't have those probiotics in the body. Like, so you basically you're taking the effect, like you're having them experience the benefit without having the microbiome actually healthy, like by feeding them.
The intention is that you're getting only the what you need to be biologically functional without giving the bacteria itself. But in practice, it's not that if I give the extract of bacteria, it's going to work and it's going to have a functional profile. Everything is so specific, and there's so many variables that it goes through. It's more about how we can do that, because the possibility, yes, you're right, this is a possibility, but how we can take advantage of this approach to get something functional, and that's very complicated.
I see.
I don't know if you still wanted to talk about the immuno-oncology, because I also have another question, but I don't know. I think
Ask your question, then I talk about immuno-oncology later.
Okay. So my question was, and this is something that has been fascinating me for a while. Maybe it's a little bit too out there, and maybe you don't know anything about this. I don't know. But I've read several times now that the microbiome affects how our brain functions. I don't know if that's, popular science and not true, but from what I've read, it can like influence our mood, our decision making, and so on. So I was wondering if you can say anything about this and how. And this gets a little bit philosophical, but it leads to the question, if what I'm saying is not bullshit, but it leads to the question, like, what is you, and is you maybe also your bacteria thinking and not just your brain? And I think that's a very interesting discussion.
Yeah, yeah. I think making this philosophical. I think it's fun. I just have to control myself, otherwise I'd be like
Yeah,
talking about this in a very philosophical way for a long time.
yeah, yeah.
And this is actually what made me fascinated by evolution at the beginning, is how much it changed the way I see the world and things. But going back to your question, um, so it's true that the microbiota influence the brain. There's what's called the gut brain axis, uh, which is a relationship between the gut and the brain, and then you add a layer of the microbiota on it, and you have the microbiota gut brain axis. Which is the microbiota plays a role. The mechanism that they can work on this can be various. It can be related with the immune system, and it can be related with neurotransmitters that that is produced in the gut. They could break, they could digest and break down some molecules that interacts with the receptors in nerve cell in nerves neuroreceptors that are in the gut, and influence how the brain perceives for example, if you're hungry, if you're not hungry, but also anxiety, depression. It changes your mood. And this is not my expertise. I don't work with the brain aspect, but one thing that you can explain causality on this, that you can use animal models, and this is in the whole field of microbiota. That's a common approach to infer causality. You can take a mice that it's more it has high anxiety, and a mice breed that is less anxious. And if you take you can develop what's called germ-free mice, where those mice they're they're born in a condition that they don't receive the microbiota from their mom when they're born, and then they are called germ-free. So they have no microbiota in their intestinal tract because they have to receive that from the environment. And then they can take the microbiota from a mouse population that is more anxious, and give to the germ-free mice. And if you take the microbiota from a mouse population that is less anxious and give to germ-free mice, you can transfer the phenotype. So the mice from a non-anxious population that receive the microbiota from an anxious population, they start to become anxious. And the mice that that is from a more anxious population receive the microbiota from a non-anxious population of mice, they become less anxious. So you can see causality on this. They specific the details of the experiments. I may not be precise because I'd have to refresh my memory in reading the papers. But this approach of making a microbiota transfer from one animal with a phenotype with another to another with a different phenotype, and transferring the phenotype with the microbiota is common and established in many fields. You can do that with diabetes. You can do that with immune oncology, like immunotherapy for responders and non-responders, for treatment, for obesity as well, and with anxiety, which would be something related with the brain. You can see those effects as an
I mean,
example.
yeah. I mean, that's just crazy. I think that's so fascinating. I
I
I
think
wonder
so.
though. I wonder though if you really can infer causality, because like I wonder if I don't know. I don't know where anxiety originates. Like it could be that the microbiome is the originator of the anxiety that causes the brain to be anxious, or maybe the brain causes the microbiome to become an anxious quote unquote microbiome, which then causes the new mouse to also become anxious. I don't know. It's like super fascinating, but it also begs the question if treatment of microbiome should be like a psychological type of treatment also. And also I think what is fascinating, just like as a funny note, is that we have these expressions like gut feeling and like
Yeah,
deciding with the gut and so on, like since
yeah,
hundreds of years probably.
yeah,
And now we're finding out that it's actually kind
yeah.
of true, even though it seems nonsensical. Yeah.
Well, because in a way, not to get too woo-woo philosophical as well, but like sometimes when you have those kind of instincts or those kind of like bodily feelings associated with your thoughts, they keep saying, uh, your thought starts as a body feeling, and then it transforms into a thought. So you could say, and that in a way that's more primordial kind of like feeling that then becomes a thought, and so you feel both in the end. you have the thought in your mind, but then you feel in your stomach something is off or something is good, because there's also the butterfly stomach butterflies, when you're like in a em- emotional in a positive way. uh
Yeah,
that's kind of like
I mean,
yeah.
yeah, the we talk a lot about meditation on the podcast, and I think Matteo and I, we've talked about this, but the feeling of being, for example, in turbulence in a storm in the airplane and being on a roller coaster, like the actual feelings are the same,
Mm.
but your brain's interpretation of the imminent danger is very different. So like one will be exciting and the other one will be anxiety inducing, even though the actual bodily feelings are the same. So I think it's our brain's labeling of some physical emotions that causes the.
But are they really the same? I would argue they're not the same, but maybe it's the thought part that like makes it different. But yeah, it's really interesting, and in a way, you could draw like a too fast of a conclusion, saying probably what you eat changes the way you think, and like your diet has a huge impact on like the way you reason and the way your mind thinks. The same as like sleeping less or more probably does, right? Um,
Yeah, yeah.
so, but it's to be considered. So in a way, one has to take care of that, right, more than one thinks, because eating like it's not just about energy and about the macronutrients, as you said, like the right vitamins and right, like, but there's also the bacteria aspect of food, which we're starting to talk about, but it's not that widespread yet. And it'll be very interesting to understand these postbiotic. I never heard of postbiotic before this podcast, so that's very interesting for me, because if one can actually inject the benefits of having like certain bacteria in your body in food, another. It's probably like, again, people think about shortcuts. It's probably even a bigger shortcuts, which not necessarily is beneficial for you, but like you get the benefit without having to even grow the bacteria in your body in a way.
But I mean, long term, you want the equilibrium in your gut anyway. So I think
Right.
it's just, yeah, it's just a trick, right?
Yeah.
If it even works. I think I have one thing. I think like we're getting close to the end, so I wanted to ask for some like maybe practicalities, because like I think a lot of this is like very scientific and very complicated. And I think it sounds like to me that Antonio, you're saying that a lot of this is like active research, so we don't know a lot of the answers that we want at this point. But are there any kind of like non-contentious pieces of advice that you can give to people, like in particular, is there anything that always, I don't know if there's even such a thing as always, but like that like almost always has a good effect, or something like, um, for example, like you talked earlier about processed food. Is there actual evidence that says processed food is actually in general bad for your microbiome? And if so, how so? And I don't know, what is like some take home advice?
Yeah. So one topic that usually is associated with a positive impact is fiber. So fiber is a complex nutrient, complex carbohydrate that they are digested slowly, and they have different type of uh, carbohydrates that feed specifically some type of bacteria. They foster the diversity of the microbiota, and in general, they are considered healthy. And processed food, you have a high fat diet compared with high fiber diet, for example. Usually, high fiber diet has a positive impact in the microbiota and also in the health of the
So
patient.
the negative effect of the processing in that case is the reduction of the fiber content.
So this is what is, at least me personally, I could say yes. It's consistent with that, but there's so many variables when you talk about microbiota in food that it's very hard for you to control every variable when you're making an analysis. So you can have thousands of complex sugar in a diet, and thousands of different species of bacteria. and then we take two extremes. Like usually when people compare, they say this one has a healthy, complex diet, and this one it's just a high fat diet, high fat, high sugar diet. And so that's different than what you'd be comparing with let's say a more balanced lifestyle. I can eat an ice cream once a while, I can eat a burger once a while, and I have my fiber, because you're just taking the extremes when you often perform this type of experiments. And then then it's more like so that you can see the signal, like you want to highlight the signal, but how much it would affect uh, something between it becomes more complicated, I would say, right? The patient population, which are like uh, real population, real life environment, they can be very diverse. Some of
Yeah.
them, but in general, high fiber would be positive, I would say. If
Yeah.
you want a general advice, like a like put a slice of tomato in your plate. Put some greens when you're eating your food. Uh, I don't know what you eat in the morning, but consider having uh, oatmeal, perhaps can have some fiber. But I'm not a dietitian, so
No, no,
I
but it's
cannot
clear.
give.
It's clear, but it's quite interesting, Antonio. I think it already the idea, like one thing to leave ourselves with, I guess, is the idea that like what you put in your body, being it antibiotics, food, what you drink as well, right? Like as a bigger impact than one thinks, I think, in terms of effect on your body responds to external stimuli, which could be like bacteria that are outside and like try to break in, or the way you even like your brain thinks. And of course, we know the energy level, right? Like if you don't eat enough, you don't have energy. That's clear. And if you don't eat certain type of food, you have less energy, right? But I think that already this combination, this kind of like balance to keep and this thing to keep in mind, I think sparks a lot of like food for thoughts, pun intended, in thinking about how you structure your days and like how you structure your habits in terms of also what you ingest. Yeah.
Yeah. There's one aspect that I think it's very interesting to highlight. So the bacteria, they're not necessarily uh, they're not deterministic of the phenotype. But they influence the phenotype, and they are responsible. It's what I would call heterogeneous response. So in similar condition, you'll have some patient that will respond very different to treatments, or in or in their body, the feature that they have. Like some eating the same amount of food, some people are going to be more likely to gain weight. Some people would keep the same weight, and you have a lot of heterogeneity. A big part of it is genetic, but often they are influenced by the environment. And the microbiota is a big environment on this. There are disease that depends on the genetic aspect of the patient, but also on the microbiota. There is one specific that cause some uh, some disruption in the vascularity in the brain, and it causes seizure in patients. And this is a disease that it requires a specific genetic background, but not everyone develops that disease. And then because of this heterogeneity, people the investigators, they are looking at the microbiota of impact, and then they model this disease in animal model, and they saw that a specific type of bacteria combined with the genetic background would induce the disease in about 80% of the mouse, the
Yeah.
mice. But without that bacteria exposure, even if they have the genetic predisposition for the disease, nothing would happen to them. And I think many brain diseases that we have, like Alzheimer, Parkinson, could be influenced by the microbiota as well. There is research on that. That's not my expertise, And the one that I'm more involved, which is in immuno-oncology. Uh, so when you work with immunotherapy, the immune system is the first barrier to treat cancer, to attack cancer in our body. So the cancer accumulates mutation, and these mutations they are translated into peptides, into proteins. They're different than our natural proteins, and the immune system can identify that. They see this cell is producing some weird proteins, so this is dangerous, and then it kills that cell. But then at some point, the tumor learns how to avoid the immune system, or the immune system starts to tolerate more the tumor. Uh, and then people receive immunotherapy. There is one thing called checkpoint inhibitors, or checkpoint blockades, that they block this checkpoint pathway in the immune system, which is something that causes tolerance, and make the immune system more aggressive. And then the immune system that is tolerating the tumor with their mutations, they start to be more aggressive against the tumor. And what is interesting is that some patients respond to this treatment really well, and sometimes they can even go to remission, which means they no longer the cancer disappears. But most of patients do not, and some of them in between. But then if you take the patients that are refractory to treatment, they don't respond to the therapy, and you take the microbiota from patients that respond to therapy, and you give to the patients that did not respond, some of them start to respond to the treatment. And this is fascinating because like a patient, it didn't work, the treatment, they were exposed, and then you say, let's try it again, but giving the microbiota from that other guy to you, and then it starts to respond. And about 25-30% of the patients start to respond again.
That's very
So
cool.
yeah, I think it's fantastic, and I could talk about a lot of the specific mechanisms on this, but I'm just giving like at least this concept and this idea, and this is validated. This is experimentally proven in clinical trials, so
That's
so
super
it does
awesome.
work. Yeah.
Super interesting.
Very interesting.
Yeah.
Antonio, really, thanks a lot. A lot of information, a lot of insight.
Hard to digest, right?
Yeah.
Yeah.
Exactly. No, but seriously, it's been a pleasure listening to you and to all these new things for me. They're definitely pretty much all new.
So we can so I'm gonna do this again, but we can digest this, and then maybe if we have more questions, we can do a second episode
Yeah.
with more specifics.